platelet plug forms first, quickest. And so it contributes to stopping the bleeding more rapidly, and then it gets reinforced by this fibrin mesh. Anybody have any questions? Now, when you take pharmacology, we will go into the coagulation cascade in a lot more detail, because then you have to study drugs like heparin and Coumadin that interact with this cascade.
If we're going into nursing, can you take your pharmacology?
No, you can't. The nursing pharmacology is entirely different. You don't actually learn drug names, and you don't really learn theory. They want you to learn classes of drugs, and simply what the purpose of that class of drug is. So for instance, you learn that heparin and Coumadin are blood thinners, but you wouldn't have to learn how they interact with the coagulation cascade. You'd have to know that one of the complications of those drugs is bleeding, but you wouldn't have to know any other details or what the antidote to that is. So it's presenting it in a much simpler fashion. So yeah, it's good for you. Okay, let's talk about some conditions associated with increased bleeding. What we call a bleeding diathesis. Platelet abnormalities. Thrombocytopenia. If you have a low platelet count, you're at increased risk for bleeding. The same thing occurs if you have abnormal platelet function, when the platelets don't adhere or aggregate. What can interfere with platelet function? Sometimes drugs can, like, what drug am I thinking of?
The blood thinning, you said?
Well, not those two. Those interfere with the coagulation cascade. But an antiplatelet drug would be like aspirin. You ever heard of patients with coronary artery disease taking aspirin once a day?
Mm-hmm.
That's to prevent platelet blood formation within the coronary arteries. Platelets interfere with, aspirin interferes with platelet aggregation. And there are a lot of pharmaceutical drugs that serve as antiplatelet drugs. And we give them to patients who have conditions that predispose toward clot formation, like atrial fibrillation, or coronary artery disease, or patients who have carotid atherosclerosis. So we want to give an antiplatelet drug to prevent clot formation at the sites of atherosclerosis. So those drugs would not reduce the number of platelets, they would simply interfere with platelet function. Now, you can also develop an increased tendency to bleed with deficiencies of one or more coagulation factors. Sometimes people have a congenital condition where they're born with a deficiency of a coagulation or clotting factor. That condition is called hemophilia. Classic hemophilia, it's also called hemophilia A, is associated with a deficiency of clotting factor 8. There's another type of hemophilia called hemophilia B that's associated with deficiency of clotting factor 9. Patients with hemophilia tend to bleed more easily, particularly when they're subject to any type of trauma. But it doesn't have to be precipitated by trauma. The most common site where patients with hemophiliacs bleed is in the joints. Why? Because the joints are subject to pounding, to wear and tear. Now, with respect to the clotting factors, we said that they circulate in an inactive form. The liver is the organ that's responsible for synthesis of almost all the clotting factors. So if you have severe liver disease, it can affect coagulation. for 4 of these clotting factors 2, 7, 9, and 10, the liver requires vitamin K to synthesize them. So these clotting factors, 2, 7, 9, and 10, are referred to as the vitamin K dependent factors. In nutrition last year, we didn't cover it this semester, a little bit of nutrition. Has anyone taken nutrition? Nobody? Well, in nutrition you learn that some of the vitamins that you consume with your diet are fat soluble and some are water soluble. The fat soluble vitamins, which are A, D, E, and K, include vitamin K. Okay? What it means is that in order to absorb vitamin K from your diet, you have to be able to absorb fat. In other words, we absorb it with the absorption of fat. And so, if you have any medical condition that interferes with absorption of fat, you're at risk for vitamin K deficiency. One such condition, for instance, is a gallstone that obstructs the common bile duct. It turns out that bile, which is delivered to the small intestine via the bile ducts, is necessary to absorb fat. So that if bile can't reach the small intestine because you have a gallstone that's blocking the common bile duct, then you have a problem not only with absorption of fat, but also with absorption of vitamin K. And so you'll develop a vitamin K deficiency, and with that, a coagulopathy. Meaning that the lack of vitamin K will interfere with production of those clotting factors, which will cause you to bleed more easily. The other day, we talked about the fact that if you have a gallstone obstructing the common bile duct, that it can lead to steatorrhea. Did we talk about that? Okay. Fatty stools.
Yeah.
And it's because bile is necessary for fat absorption. So if bile can't reach the small intestine, you're not going to absorb fat. It's going to pass into the large intestine and out the body to the feces. We call that condition steatorrhea. So many patients with steatorrhea also bleed or bruise easily because they're not absorbing vitamin K. Again, its absorption is dependent upon the absorption of fat. So you can develop a deficiency of coagulation factors due to inadequate synthesis or absorption of vitamin K. Some drugs purposely interfere with vitamin K, or the liver's use of vitamin K. An example would be warfarin. It's also called Coumadin. Okay. It interferes with the use of vitamin K to synthesize those clotting factors, 2, 7, 9, and 10. So it makes people bleed more easily. Coumadin is the same stuff we put in rat poison. Okay. So Coumadin would be an anticoagulant drug, just like heparin would. We give some people anticoagulants to help prevent them from clotting. Many times it's because they've had a heart attack or a stroke caused by a clot. And we want to interfere with clot formation. Now, Any questions about either platelet plug formation or coagulation? Nobody? No good on this? Let me go ahead and finish up the blood typing then, because this is easy. Okay? Blood typing is based upon markers that are present on our red blood cell membranes. These markers are glycoproteins, proteins with a little sugar chain attached, okay, at the surface of the cell membrane. And there are three types of markers that you might see on red blood cells. And when I'm talking about these markers, I'm talking about specifically the ABO antigens. The term antigen means it's usually a protein that serves as a marker that our immune system can recognize. Okay? These are self-antigens because they belong to self. So the major antigens, the major markers on the surface of red blood cells are of the ABO type. Now, are there other types of antigens present on red blood cells? Yes. But the ones that we're most concerned about are those ABO antigens. Okay. So some people have A antigens present on the surface of red blood cells, and they have a particular size and shape, which is going to represent them as little triangles. Okay. And these antigens, by the way, are coded for by a pair of genes, one of which comes from mom, the other comes from dad. Other folks have B antigens. They look different. We'll represent them as little circles on the surface of their red blood cells. Now, if you received the gene that codes for A antigens from one parent and the gene that codes for B antigens from another parent, then guess what? You're going to have both types of antigens on the presence of your cells. Okay. So in this case, type A blood, type B, but if you have both antigens, you'd be type AB. And then some folks have genes that do not code for either A or B. Okay. You say they have type O blood. Now, these two genes, one of which is from mom, one from dad, are considered codominant. That means each of the genes is going to express its product. If you have the A gene from dad, you're going to have A antigens on the surface. If you have the B gene, it's going to express itself. If you have both genes, both will express themselves. So these genes are considered codominant. Okay? Now here's the interesting thing. If you're born with the A antigen, you're also born with antibodies against B. So you're born with anti-B antibodies. If you're born with the B antigen on the surface of your red blood cells, you're born with anti-A antibodies. Now, if you're born with type AB blood, are you going to have any antibodies? No, because otherwise they would attack self, your own red blood cells. So no antibodies. On the other hand, if you're born with type O blood, you're born with both anti-A and anti-B antibodies. Now, how do we find out what type of blood you have? Well, we do a type of test called blood typing. We use a particular fancy type of slide, glass slide, that has 2 wells in it. And in one of the wells, now in both of those, in both of the wells, we put a drop of the patient's blood that we're typing. We do a drop of blood in both wells. And in the first well, we're going to mix that drop of blood with anti-A serum. This is serum that simply contains antibodies against the A antigen. And in the second well, we're going to mix the drop of blood with anti-B serum. Again, simply antibodies against the B antigen on the surface of red blood cells if they exist. Here's what happens, by the way, if you mix type A blood with anti-A serum. Antibodies look like this. They're Y-shaped molecules that have arms that bind to antigens. Okay? And anti-A antibodies are going to have a configuration at the end of the arm that's complementary to the antigen that it's binding. Okay? So, because there are 2 arms to each antibody, these antibodies can cross-link red blood cells. So that means you might have another antibody here.